In this section of our notes, we're going to talk about the pGlo lab and what exactly it's going to demonstrate for you. This pGlo lab shows you that geneticists and even high school students in a lab can transform bacteria. And we can do this by using methods like the heat shock method and using chemicals like calcium chloride to increase our efficiency of transformation. That heat shock method and the addition of calcium chloride makes the cell membrane of bacteria cells more permeable, so they're more likely to take up plasmids. Now, ultimately, what we're doing in this lab is we're inserting DNA so that bacteria can clone it for us. So this lab is showing you that we can clone DNA by inserting it into bacteria cells. Before they divide, they'll copy the plasmid. So ultimately, we'll end up with billions of bacteria cells and billions of these plasmids. So we can clone DNA by inserting it. But we do know now that we're more likely to use PCR to make multiple copies of DNA instead of bacteria. This lab also shows you that we can control gene expression in these transformed engineered bacteria cells. We can put specific parts into these plasmids that we're going to insert, and that allows us to control whether bacteria are expressing genes and making proteins or not. Now let's take a look at the different parts of this pGlo plasmid that we're going to insert into E. coli. We engineered a recombinant plasmid a couple class periods ago. It was very simple. We inserted an insulin gene and an AMP-resistant gene. But in this pGlo plasmid, there's a lot more to it than what we had engineered in our simple model. First of all, let's make sure you understand what this part of the DNA would do. This is called the origin of replication. It's just a sequence on DNA where replication would begin. That means that part of the plasmid, when the cell goes to divide, replication is going to start there. Helicase is going to unzip the plasmid there originally for replication to take place. And again, if we want bacteria to make multiple copies of DNA for us, then they have to have the ability to replicate the plasmid. So we have multiple plasmids over time. The second gene I want to talk about is this one right here, that beta-lactamase gene. Genes code for protein. So this gene codes for a protein called beta-lactamase. And it's a protein that destroys ampicillin, but let's put cleaves. It basically breaks up that ampicillin molecule, which no longer allows it then to kill the bacteria cells. So any bacteria cell that has a beta-lactamase gene, that cell is ampicillin resistant. So that gene is going to give the bacteria ampicillin resistance. Now, you need to know why we would give them ampicillin resistance, and it's simply because we're not going to transform all bacteria cells. And this is going to allow us to select for the ones that were transformed and get rid of the ones that weren't transformed. The next gene we're going to look at is this green fluorescent protein gene. And this gene codes for a protein that glows whenever we shine UV light on it. So any transformed colonies or transformed bacteria cells, they're going to glow green under a UV light. Now there's two different reasons why Bio-Rad put this in this plasmid, to show us some different things related to what we can do with DNA. One is that by putting this plasmid in here and putting this part that we're going to go over, they're showing us that we can turn on and turn off genes. Because you're going to see in some plates, the bacteria cells are going to be making the green fluorescent protein. That gene's going to be turned on. But in other plates, they're not going to be making the protein. That gene is going to be turned off. In addition, I want you to know that by adding this gene, this helps us look for or select those organisms that were transformed. We can easily identify the transformed from the non-transformed bacteria because the transformed are going to be green and the non-transformed are going to be white. We use the GFP protein a lot in research. When we genetically modify organisms to have human diseases, then lots of times we also will insert this gene for GFP. So we can easily look at our organisms that we're doing research with, and we can tell the ones that are green, the ones that glow, are the ones that were transformed. They have the genetic disease, from our control group organisms that do not have that GFP protein. So that's our control group. Now, related to the GFP gene is this area of the DNA that's inserted right there before that GFP gene. And this area is a promoter, and it's an operator. We talked about those when we were doing our models of operons. We know that operators can have repressors bound to them, and that can turn off the genes that are downstream. And this specific operator that's inserted, or the gene for the operator that's inserted, is affected by a sugar called arabinose. When you think of arabinose, I want you to think of lactose and what lactose did with the lac operon in those models that we built. Arabinose has the ability to bind to the repressor that we're going to talk about here in a second. So again, that part of the pGlo plasmid is put in there to show you, so that we'll be able to show you that we can turn on or turn off specific genes. And in this plasmid, it's that GFP gene that we're going to regulate using a different molecule like arabinose. And then finally, if we want to turn on and turn off genes and use promoters and operators to do that, then we have to have that gene that codes for the repressor protein that can bind to the operator. So again, we just have to put the pieces in there so that we can effectively turn on and turn off genes in this lab. I rewrote this plasmid to look like this when I was explaining that model to you, or I'm going to explain this model to you. And it's showing you that we can show DNA and these recombinant plasmids different ways. So this is the exact same thing as this. I just made it linear, but you can still see there's an area of DNA that's the origin of replication. I have a BLAC gene that codes for AMP resistance. I have the operator here right in front of that GFP gene, which can code for the green fluorescent protein. And then finally, that regulatory gene, which will code for a repressor. So let's make sure you understand exactly what arabinose is going to do so that we can see genes being turned on or turned off in this lab. Arabinose, again, it's a sugar, and I put it in the Petri dish. So here I have a cell, and the cell has the plasmid. This is just another way to model the different parts of a plasmid. And this cell is growing in a Petri dish without arabinose. So that sugar is not present. Then this is what would be going on in that cell. It has a regulatory gene, so it has made a repressor protein, and that repressor protein is made active, so it binds then to the operator. RNA polymerase, when it binds to promoter, it is stopped by that repressor. So therefore, that GFP gene is not being expressed. It is off. Some bacteria cells that are transformed are going to be put on an agar plate with arabinose. And just like with lactose, arabinose is going to move into the cell, and it would bind to that repressor protein right there, and it's going to make it inactive. And we see that right here. Here is that inducer, or the arabinose. It is bound to the repressor. The repressor is no longer bound to the operator. So that GFP gene is being expressed. It is being on, and that cell is ultimately making GFP, green fluorescent protein, which will then cause that cell to look like it's glowing. So no arabinose, no GFP produced. When there is arabinose, then we're going to get that GFP protein produced. So again, this lab is just showing you that we can engineer plasmids that can have control mechanisms, that we can control whether certain genes on the plasmid are going to be turned on or off just with manipulating molecules in the environment, like sugars. Now we're going to look at the general layout for this lab. You're going to be given two different vials, and I'm going to give you the plasmid that was engineered by Biorad. And we're going to take some of the bacteria that we are growing in class. So you might indicate that the bacteria we grew. And that was to help you practice some of these techniques related to growing bacteria. But remember, the plasmid was from Biorad. So if you want to put from Biorad, so you understand, we didn't engineer this recombinant DNA. We're just simply going to try to insert it or transform bacteria cells with it. On one of the vials that I give you, you're going to write plus pGlo. And that simply indicates that we're going to put the plasmid in this vial, and we're also going to put our bacteria cells that we grew. On the other vial, we're going to write negative pGlo. And that means that we do not add plasmid to these bacteria cells. We're just simply putting our bacteria cells into this vial. Now, both of these vials are going to be heat shocked because we have to keep that constant between these two samples. So we're going to heat shock this one and this one. Remember, it's the heat shocking that is going to make the plasma membrane of these bacteria cells a little more permeable and hopefully allow some of these plasmids to move into the bacteria cells. So let's indicate that we have some free plasmids in there. And let's show some of these plasmids transforming the bacteria, which means that they're going through the cell membrane and into the bacteria cells. In this sample over here, like I said, we're going to heat shock them, but there's no plasmid or extracellular DNA to enter or transform these bacteria cells. Now, when we're doing the heat shock method, basically what we're doing is we're taking this vial, we're going to put it on ice. So it's going to make the solution very cold. And then very quickly, we're going to transfer it to a hot water bath and keep it there for a few seconds. And then very quickly, we're going to transfer it right back to a cold water bath. And that's basically all that the heat shock method is. Now, before we heat shock them, we're also going to add some calcium chloride because that's a chemical that helps that cell membrane become more permeable, which might allow some more extra pieces of DNA into the cell. So we're going to put calcium chloride in both of these vials. Okay, from there, what we're going to do is we're going to take two samples from this vial, and we're going to put them on two different petri dishes that are full of agar. And we're going to take two samples from this vial and put them on petri dishes as well. Now, I'm going to be setting up the petri dishes with you, so they're already going to contain the agar and the necessary chemicals for each plate. And they will have labels on them like this. It is critical that you understand what these labels mean. So let's talk about what I'm going to do to set up these 4 different plates. First of all, you're going to notice that in every single one of these plates, you see this term, which is LB. LB stands for nutrient broth, and that's just simply food for bacteria cells. Bacteria cells need food just like you and I do. So you can see that all 4 of these petri dishes are getting food. So let's indicate that. So this one's getting food. I'm going to write down here. This simply means food, food, and food. Now on this first plate, you see the next thing that's listed is AMP. AMP stands for ampicillin. And remember, ampicillin is an antibiotic. So this plate, I'm also going to add an antibiotic to the agar before it solidifies. And you can see I'm doing that to this plate. So let's indicate this one's getting the antibiotic, and this one's getting an antibiotic as well. And this one is not getting an antibiotic. The next label you see here on this first one is ARA, and that stands for arabinose. Remember, arabinose is the sugar. You can see that this is the only plate that's getting arabinose. And then I want to point out this one's labeled plus pGlo because I'm going to put the bacteria from this vial, and these are the bacteria that have been exposed to the plasmid. So let's think about what's going to happen to these transformed and non-transformed bacteria on each of these different plates. You can see here in this bacteria cell that I have shown this cell has been transformed. So it has that pGlo plasmid. And you can see a cell here that does not have the plasmid. Remember, the plasmid contains the gene that codes for ampicillin resistance. So if this petri dish has ampicillin on it, then this bacteria cell is not going to be able to survive. It's going to die. Same with this one right here. This is a non-transformed cell. These two are going to be able to survive because they have that ampicillin resistant gene. Moving over to the next one, we can see that we have two non-transformed bacteria cells in here. And since this plate has ampicillin in it, then that means that these two will not be able to survive. But the other two that were transformed will. On this third petri dish, you can see that none of the cells have been transformed because we didn't expose them to the plasmid. And we should see every single one of these cells should die. And then on this last plate, you can see that, again, there's no transformed cells, but all they are being grown on is agar that contains food. So they should be fine. There's no reason for them to die. So let's take a look at these examples and really what you're going to see whenever you're looking at these petri dishes. In this first plate, we know that if we've transformed any of the bacteria cells, then they're going to live because they have the gene that codes for ampicillin resistance. So we should see the bacteria growing. So we should see some growth. And remember, this plate contains arabinose. It's the sugar that's going to turn on the transcription of the GFP gene coding for this green fluorescent protein. So when we put a UV light over this petri dish, we should also see these bacteria cells glow. In the next petri dish, we should see surviving bacteria colonies because, again, those that were transformed have the resistance for ampicillin. So we should see growth. But the only way to turn on those genes, or that gene that codes for GFP, is if arabinose is present in the agar, and it's not. So these colonies should not glow. In this third petri dish, we should have absolutely no growth. This dish has ampicillin, so only cells that have taken up a plasmid that has a gene that codes for ampicillin resistant would be able to grow, but these cells weren't exposed to the plasmid. And then finally, we're taking bacteria cells and we're just growing them on agar with ample food in a warm location. So there should be plenty of growth. So we should see lots of growth in this last petri dish. Whenever we're done, we're going to end up counting the number of colonies on this plate. And then we're going to follow some steps, and that's going to help us calculate what's called the transformation efficiency. Basically, this is how well you heat shocked. Or this is like a grade on how well you transformed your bacteria cells. So when we're done making these calculations, if we get a calculation like 25%, then that means that we transformed 25% of the cells that we mixed in there with the plasmid. So that means 25% of the bacteria took up this plasmid or were transformed. In general, this is what you should learn from this pGlo transformation lab, is that we can take DNA from one organism and we can insert it into a plasmid or DNA of bacteria cells. Because the central dogma and the genetic code and the structure of DNA is the same in all organisms, then those bacteria cells should be able to make these proteins that other organisms have the genes for and can synthesize. You should understand that we can put selective markers in the plasmid so that we can identify which cells are transformed and we can isolate them from the non-transformed cells. This also shows you that we can manipulate plasmids, so we can turn on or off the production of proteins that are coded for by genes that we inserted on the plasmid. And we can also manipulate the environment in the lab so that we can increase the transformation efficiency. So we can use the heat shock method, and we can use chemicals like calcium chloride, which makes the cell membranes more permeable.